The idea of fixing a damaged brain, whether it's from a stroke or a disease, it's been one of the holy grails of medicine for forever, really. And the big hope has always been cell therapy. The idea is simple, right? Just put new, healthy cells in to replace the ones that were lost. The only problem? The brain doesn't give up its secrets easily.
So, it all comes down to this one question. A question that drives billions in research funding and the hopes of millions of people. Can we actually use new cells to fix the most complex object we know of? For a long time, the answer has been a shaky maybe, because it's always been held back by this one massive, persistent problem.
And here it is. This is the number that's been holding everything back. 3%. In so many of these groundbreaking studies, that's the percentage of transplanted cells that actually survive. Just 3%. Think about that. That's like trying to build a skyscraper when 97 out of every 100 bricks just turn to dust. That's what scientists have been up against. So why? Why is the number so low?
Well, it's because for the longest time, the whole process has been a complete black box. Researchers would inject these precious cells into the brain and then what? They just have to hope for the best? They had no way to see what was happening after the injection, no way to know what happened to those cells. They were flying blind.
So when you can't see what's going on, it's hard to fix things. And it turns out, it's a disaster on multiple levels. The injection itself can literally tear the cells apart. A huge number of the survivors just leak right back out. And the few that manage to stay, they get thrown into this hostile, inflamed environment and die. But the real kicker, the problem that connects all of this, is that you couldn't watch any of it happen.
And you don't have to take my word for it. The researchers said it best themselves. This whole phase after the transplant is a mystery, a black box just waiting for someone to turn on the lights. Well, it looks like someone finally flipped the switch.
And that's where this incredible new framework comes in. It's called OPTRACE, and it's a breakthrough designed to do exactly that. Turn on the lights. It's not just one little fix, it's a whole new system to guide, track, and finally understand what's going on with cell therapy. Now, OPTRACE, I know, it's a bit of a mouthful. It stands for Optical Imaging Guided Transplantation and Tracking of Cells. But what it does is actually really simple to grasp. Think of it in 2 steps. Step 1, use light and imaging to guide the cells in perfectly. Step 2, use that same idea to track each individual cell over the long term to see what it's really up to.
So, if we want to boil it all down, it's like this. OPTRACE gets rid of the guesswork. You're swapping a blindfold for a high-definition surgical camera to do the procedure. And then you're attaching a tiny, unique GPS tracker to every single cell so you can follow its journey afterward. It's incredible. All right, so let's dig into that first part, the guiding. How exactly do you use light to turn this blind procedure into something so incredibly precise?
Okay, just look at this comparison. The difference is night and day. The old way, a clunky, opaque metal needle. You couldn't see anything. But the OPTRACE way, it uses a tiny, transparent glass pipette. You can literally watch the cells flow into the brain in real time, making sure they land exactly where they need to with way less damage. It's a total game changer. Okay, but wait, there's a catch. This amazing new precision creates a new challenge. To see everything with the microscope, you have to inject the cells really close to the brain's surface. But as we just talked about, that's where they leak out the most. So, what do you do? How do you get them to stay put?
And the solution they came up with is just brilliant. It's called the pulse elevation technique. Instead of one big shove of cells, they do this little dance. They inject a tiny pulse, pull the pipette back just a fraction, inject another tiny pulse, and on and on. This actually creates little pockets in the tissue for the cells to settle into, which means way more of them stay right where you want them.
But it gets even cooler. OPTRACE isn't just about what happens during the procedure. It's about using math to figure out the perfect plan before you even start. So their first mathematical model pretty much confirms what you'd guess. It shows that the deeper you can inject the cells, the better the surrounding brain tissue creates a seal. No more leaks. This lets them find that perfect Goldilocks zone, deep enough to keep the cells in, but shallow enough to still see everything with the microscope.
Now this next one. This one is completely wild, totally counterintuitive. You'd think, hey, let's put in a big batch of cells, right? More is better. But no, their model proves that's the worst thing you can do. A big clump of cells means the ones in the middle can't get any oxygen or nutrients from the blood. They basically suffocate. So the real secret, smaller, more targeted injections are actually way better for survival.
Okay, so we've nailed the delivery.
We've got more cells staying alive right where we need them. Fantastic. But now for the second part of the miracle, finally opening that black box. How do we see what these cells do over the next days, weeks, and months?
The answer is this amazing labeling strategy they developed called ICAM. It's basically a way to give every single cell its own unique, colorful barcode before it's injected. They do this with a mix of fluorescent proteins, so each cell lights up with its own special color combination. It means, for the first time, you're not just watching a faceless crowd, you can follow every single player on the field.
And just like that, the black box becomes a living movie, and we get a front row seat. You can watch the whole drama unfold. On day 1, the local brain cells are totally stressed out, like who are these new guys? Then, by days 2 and 3, the brain's own immune cells, the microglia, swarm in. They're the cleanup crew, checking things out. And by day 5, you can see things start to calm down as the new cells begin to integrate. We are literally watching the brain heal in real time.
So we can get the cells in better and we can watch what they do. That's incredible. But let's bring it all home. What does this all really mean? Why is this such a monumental step forward?
Well, what this technology unlocks is just huge. 1, we can finally do something about that awful 3% survival rate. 2, we're doing far less damage to the brain in the process. 3, and this might be the most important part, we can finally figure out why things work or don't work. We're learning the rules of the game. And when you put all that together, it means we can speed up the development of real cures for some of the most devastating diseases out there.
At the end of the day, what OPTRACE really does is take that scary, unknowable black box and turn it into a living microscope. It gives us this incredible window into the healing process, letting us watch that complex dance between new cells and the brain so we can finally learn how to help the brain do what it wants to do, heal itself.
And that leaves us with this final, tantalizing thought. For the first time, we've moved past just hoping for healing. We can now see it. We can watch it happen. So the question is no longer if we can repair the brain. The real question is, how well can we do it? Now that the lights are on, what are we going to build?